So as you all know, CAR T-cell therapy with autologous anti-CD19 CAR T-cells has completely changed the way we currently treat patients with relapsed or refractory large B-cell lymphoma. There are 3 products currently approved by the FDA in the United States as third-line option. One of them is Axi-cel, along with Tisacel and Lisa. And Axi-cel is also the autologous CAR T-cell product currently FDA as a second-line option for patients with refractory large B-cell lymphoma. Currently, with the use of single-agent Axi-cel, either in second or third line, the chance of cure is close to 40%. So there's a lot of interest to try to combine Axi-cel with other to further improve its efficacy overall outcomes in patients with such an aggressive condition, such as relapsed or refractory large B-cell lymphoma. Preclinical data show that combining Axi-cel with an agent targeting antigens other than CD19, such as Rituximab, a monoclonal antibody targeting CD20, may favor overall CAR T-cell activity due to upregulation of CD19. So this was the preclinical rationale for ZUMA-14, which is a combination trial, a phase 2 trial investigating the efficacy of Rituximab in combination in patients with relapsed or refractory large B-cell lymphoma. In this study, Rituximab was given initially during the lymphodepleting conditioning chemotherapy, so on day -4. Typically, the conditioning goes from day -5 to days -3. And on day 21, and then subsequently every 28 days for a total of 6 infusions. And the primary was response rate. Overall, we were able to enroll 26 patients across multiple sites. These were heavily pretreated patients, refractory, and with very high tumor burden. We all know being in a favorable condition in terms of either increased toxicity or decreased efficacy for CAR T-cell therapy in patients with large B-cell lymphoma. Despite this, we first of all saw that the safety profile of Axi-cel was not negatively impacted by the combination with Rituximab. A rate of CRS of any grade or high grade and the rate of ICANS of any grade or high grade was comparable to a tumor burden matched population of patients treated on ZUMA-1. And most importantly, we did not see any increase in the rate of infectious complications, either any grade or high grade. The other thing that we were able to observe is the overall CAR T-cell amplification when Rituximab was used in combination didn't seem to be negatively impacted. And this translated in very good efficacy data with the overall response rate higher than 65%, also with very prolonged follow-up, now close to 2 years. This is very promising. Of course, it's just the first step. And it's really a proof of concept. They're trying to target antigens other than CD19 during CAR T-cell infusion, such as in this case, during lymphodepleting chemo, and then several times after CAR T-cell infusion, may overall improve CAR T-cell efficacy without impacting safety. And this, of course, is paving the way to new strategies such as CAR T-cells with double targets, targeting both CD19 and CD20, or multiple CAR T-cells combined in a single product. Most of these trials are already ongoing, and we all look forward to their readout in the next few years.